A number of people ask a question as how to create a software in Python or how to write a script in Python that can convert a DNA entry from the user to a reverse complement of that DNA. So this video is meant to answer that question. To start with, we'll need to have a variable that contains user input. For instance, we write down DNA equals to input, and then write down in a string, please enter your DNA sequence below. Then write down a new line mark so that it asks the user in the next line. So what happens here is that this line of code asks for user input, and the user should input a DNA string or a DNA sequence below. The user has no idea as to whether write it in lowercase or uppercase, so we've got to dictate it later on. This is an example that shows a toggled upper and lowercase DNA entry from the user. Then press enter. Next, what we've got to do is to convert this into uppercase values. So an easy thing to do is to have another variable, let's say DNA underscore upper, that contains the first in uppercase fashion. So write DNA.upper, open and close parentheses, then it saves the entry in uppercase values. So you can print out the second variable so that you can see what happened. So write down print, open and close parentheses, the second variable, which is DNA underscore upper. Press enter, and you can see that the variable became uppercase values, so it contains uppercase values. Now we need to reverse this one. To do so, we write down the variable containing the second variable with 2 colons and -1. This makes the DNA sequence reversed. When you press enter, you'll get the values in a reverse manner. So you can also print out this variable so that you can see it. And you can concatenate it as well, so that you tell the user that this is the reversed DNA sequence or DNA strand. Then show the user. This is the reversed DNA sequence. If you have a look, the TAA is at the end. Now it became AAT, which is the reversed TAA, as you can see here. If you have a look at the end, it is TGA, which is the reverse of AGT. Since we need reverse complementation, we need to have a for loop to show the values and compare them to the if statements in the for loop. So what we've got to do is to write down for every occurrence of adenine, let's say. So for I in the reverse DNA sequence, if the value of I is equal to adenine, we've got to convert it and write down, let's say, print T. So if I is equal to A, print T. If I is equal to T, print A. The same goes for C and G. So if I is equal to C, print G, and if I is equal to G, print C. If you have a look, the other if statements are written in LF, which is an easier way of writing if statements one after the other. If you execute this code or this code block, what you've got to understand is that it shows the values one after the other in different lines. So the first value is shown in the first line and the second in the second, and so on. So TTA is the reverse complement of AAT, and so on. Sometimes you would like to get the values in a single line because you supply it in a single line and you want to get the values in a single line also. So you can change the for loop slightly, as I'm showing it here. For I in DNA underscore upper underscore reversed, you write down the same if statement. If I is equal to A, print T. However, we add end equals to an empty string. So it changed the ending of the empty strings. Now you get the values in a single line. I hope you enjoyed today's session. Thank you.